Hello, medicos! Welcome to the Physiology series. What's something people don't know about the eye? At the heart of this intricate system lies a remarkable structure, the lens. This transparent biconvex marvel acts as a biological magnifying glass, bending light rays to focus them precisely onto the retina, the eye's light-sensitive inner lining. The lens's ability to refract light is measured in units called diopters. Diopters quantify the lens's optical power, indicating how strongly it converges or diverges light. A higher diopter value translates to a greater ability to bend light. Think of it like this. A lens with a higher diopter value acts like a more powerful magnifying glass, bringing objects into focus at a shorter distance. The lens's primary function is to ensure that light rays entering the eye converge at a single point on the retina, creating a sharp, clear image. The distance between the lens and this focal point, where the image is formed, is called the focal length. This precise focusing ability of the lens is crucial for clear vision. The retina, a thin layer of tissue lining the back of the eye, is the stage where light is transformed into electrical signals, the language of the brain. This intricate neural tissue is a marvel of biological engineering, composed of multiple layers, each with its unique population of cells. The journey begins when light passes through the transparent layers of the retina, reaching the photoreceptor cells located at the back of this cellular tapestry. These light-sensitive cells, known as rods and cones, are the true transducers of the visual system, converting light energy into electrical signals. Rods, responsible for our ability to see in low light, are highly sensitive to light but cannot distinguish colors. Cones, on the other hand, operate in brighter light and are responsible for our color vision. Emerging from the back of the eye, the optic nerve serves as the vital link between the retina and the brain, transmitting the neural impulses that carry visual information. This cable-like structure, composed of millions of nerve fibers, carries the language of light, translated into electrical signals, to the visual processing centers of the brain. As the optic nerve exits the back of the eye, it travels through a bony opening in the skull called the optic canal. It then meets its counterpart from the other eye at a junction known as the optic chiasm. At the optic chiasm, nerve fibers from the nasal side of each retina cross over to the opposite side of the brain, while fibers from the temporal side remain on the same side. This partial crossing of fibers is crucial for binocular vision, allowing the brain to combine information from both eyes to perceive depth and three-dimensional images. Imagine our eyes as cameras, capturing the world around us. In this intricate biological camera, photoreceptor cells act as the film, sensitive to light, and responsible for converting light energy into electrical signals that the brain can understand. These specialized cells, located in the retina, come in two main types, rods and cones. Each type plays a unique role in our visual experience, from enabling us to see in dim light to perceiving the vibrant spectrum of colors. Rods, the more numerous of the two types, are incredibly sensitive to light, allowing us to see in low-light conditions. They excel at detecting motion and discerning shapes and outlines, but they cannot distinguish colors. Cones, on the other hand, operate in brighter light and are responsible for our color vision. They are less sensitive to light than rods, but provide higher visual acuity, allowing us to see fine details and perceive the world in vivid color. At the heart of photoreception lies a remarkable molecule called rhodopsin, the key protein found in the rods of our retinas. This light-sensitive molecule acts as the initial receptor for light, playing a crucial role in converting light energy into electrical signals. Rhodopsin is a complex molecule consisting of two main components, a protein called opsin and a light-absorbing molecule called retinal. Retinal is derived from vitamin A, highlighting the importance of this vitamin for maintaining good vision. When light enters the eye and reaches the retina, it strikes the rhodopsin molecules within the rods. This interaction triggers a series of changes in the retinal molecule, causing it to change shape. This shape change, known as isomerization, initiates a cascade of molecular events that ultimately lead to the generation of an electrical signal. The electrical signal generated by this process then travels through the layers of the retina, eventually reaching the ganglion cells, whose axons form the optic nerve. Phototransduction, The process of converting light energy into electrical signals is the fundamental principle underlying our sense of sight. This remarkable process takes place within the photoreceptor cells of the retina, specifically in the outer segments of rods and cones, where light-sensitive molecules reside. The process begins when a photon of light strikes a rhodopsin molecule in a rod cell. This interaction triggers a conformational change in the retinal molecule, activating the opsin protein. Activated opsin then activates a protein called transducin, which in turn activates an enzyme called phosphodiesterase. This enzyme breaks down a molecule called cyclic GMP, or cGMP, which normally binds to and keeps sodium channels open in the rod cell membrane. As cGMP levels decrease, the sodium channels close, reducing the influx of sodium ions into the rod cell. This closure of sodium channels causes the rod cell to hyperpolarize, meaning its membrane potential becomes more negative. The change in glutamate release is sensed by the bipolar cells, which then transmit the signal to the ganglion cells, the output neurons of the retina. Rods, sensitive to dim light, provide us with night vision and motion detection, while cones, responsible for color vision, allow us to perceive the world in all its vibrant hues. Signals from the rods and cones are then transmitted through the layers of the retina, reaching the ganglion cells, whose axons bundle together to form the optic nerve. The optic nerve carries these electrical signals, carrying the language of light to the brain. At the optic chiasm, nerve fibers from the nasal side of each retina cross over to the opposite side of the brain, allowing for binocular vision, our ability to perceive depth and three-dimensional images.